Good morning or good afternoon. Welcome to the next in a series of webinars from NIIMBL, the National Institute for Innovation in Manufacturing Biopharmaceuticals. My name is Chris Roberts. I'm the Associate Institute Director with NIIMBL, and I'm going to help moderate the session and also present some of the work. I'm joined today by my colleagues, Dr. Barry Buckland and Dr. Jen Mantle, who will also be co-presenting as we go through the webinar today. For those who haven't maybe attended one of our recent webinars, this webinar is being recorded. You'll be muted throughout the presentation as an attendee. You can use the chat feature at any point in time to ask a question or post a comment, and we'll be able to see that, and then I'll help to curate that at the end when we have the Q&A. This webinar is open to NIIMBL members and non-members. So for our NIIMBL members who are used to looking at webinars or participating in webinars that are NIIMBL member only, please do not post confidential or proprietary information in the chat. This webinar is not covered under NIIMBL confidentiality. There will be a survey that will be sent out. So anyone who registered as an attendee, you'll receive a survey link by email soon after the webinar finishes. If you complete that survey, it'll give you immediate access to the webinar recording. And so that will help give you that as a nice feature at the end. So with that, I'm going to hand over the first part of the presentation to Barry. And so Barry, over to you.
Yeah, thanks, Chris, for the introduction. And go to the agenda slide. So today there'll be a brief overview about NIIMBL itself, for those of you who are less familiar. And then we'll cover both the current status of where we're at with the NIIMBL-led vaccines program, as well as our future plans. So that's the high-level version of the agenda. Next slide, please. And we're very grateful to the support we've received, both from NIST and also from the Global Health Fund via the Bill and Melinda Gates Foundation. And NIST have been terrific partners with us and have helped us a lot as we progress. And the Bill and Melinda Gates Foundation have a special thank you because they really helped us start up our program in vaccines about 5 years ago. Next slide. So the NIIMBL mission is to accelerate biopharmaceutical manufacturing innovation and support the development of standards, and also educate and train a world-leading biopharmaceutical manufacturing workforce. And so the workforce part was really with guidance from the Department of Commerce as one of their main interests. And when we started, that was something that was really of perhaps lesser priority to begin with. But what we've learned is actually this workforce development component has been of great interest to our members. And I'd like to really thank John Balchunas for leading us in that whole activity. The next slide. These are the Manufacturing USA Institutes. NIIMBL is one of those. You'll see in the middle column of biomanufacturing. And NIIMBL was started at it was really at the end of the Obama administration and has been running for 6 years. In the beginning, and the very beginning, we had funding, but no members. And so the growth you've seen at NIIMBL has all occurred during that time. So the next slide, please. This is the high-level view of the model. The Manufacturing USA Institute, and NIIMBL being one of those, is in the center. you'll see that our members embrace academia, national labs, industry, both large companies and small. And also in the bottom part, the red, shown in red, is the government, different agencies in the government, and both federal, state, and local. And the activities include applied research, technology development. It would also include demonstrations of manufacturing and technology workshops. And what you'll see on the left column, the federal funding with 100% co-investment, that's the model. So whatever money we have from the federal government, we match with contributions from industry and primarily industry. The next slide. So this is the funding to date. It's both from NIST and also non-federal sources. As I mentioned, we have matching funds for the federal money. And so the next slide illustrates all of our members. And so as I mentioned, when we started, and it was really driven, this initiative of NIIMBL, driven by Calvin Lee at the University of Delaware. But when we started, we had funding, but we didn't have members. And so this, we're very delighted, the growth of membership within NIIMBL. And this was as of August 2022. It's actually grown a little since then. And from a vaccine point of view, very happy that most vaccine companies are represented within NIIMBL. And going forward, this is actually a critical point for us at NIIMBL. Next slide. So when we started NIIMBL, we had a project called Process. Ideas were initiated by members. There were collaboration across organizations. Typical project was an academic working with a small company, working with a large company. And the priorities were established by industry, and the selection was driven by our industry members as to which projects to support. We're continuing with the project core process. We've evolved, because one of the things we realized with the project core process that they would fit under various umbrellas. So, for example, process intensification included quite a few of the projects that emerged from the project core process, and the same with big data. And so those have emerged as NIIMBL-led programs where we have a theme, such as process intensification, big data, in this case, vaccines, viral vector would be another one. And there we have a longer-term vision and ambition, and aligned to specific business goals. We work a lot with our partners within NIIMBL to determine the content of these NIIMBL-led programs and seek significant engagement by industry, both to choose the topics and also to evaluate which ones to support. Next slide. So this is the NIIMBL-led projects to date. We have large companies work together. This is a very important part of the process to define and shape the program. And then as we move forward, structure it in such a way to get direct engagement amongst industry leaders through a steering committee, and include small companies and academic partners to advance specific scopes of work. Next slide, please. So I'm going to hand it over to Chris, who's going to talk about current funded projects, and both previous and current. So over to you, Chris.
Thank you, Barry. So our NIIMBL portfolio, if you look both historically and current active projects, is over 40 projects in the vaccine space, or the vaccine manufacturing space, I should say, funded by a number of different sources, including our cooperative agreement with NIST that supports many of our activities for NIIMBL, not just the vaccine space. The CARES Act, funds from the American Rescue Plan Act, and the NIIMBL Global Health Fund with BMGF, as Barry pointed out in an earlier slide. We have 18 active projects with another group soon to be launched through the NIIMBL Global Health Fund through our recent project call. When you look at some of the statistics, that gives us over 60 different organizations, project participants from academia, small and large industry, nonprofits, and federal stakeholders. Seeing sort of a graphic over here on the left, where you can look at some different topic areas, depending on how you might want to parse the space of what the projects focus on, and some of them do overlap in more than one space. But we have significantly invested in test beds, things to help with advancement of formulation and stability for vaccines, workforce and public health, or workforce training, I should say, and workforce and training in public health, analytics and process analytical technology, bioprocesses, and beyond. It does include topics across the relevant vaccine types in this space and reagent targets. Just a few are listed here, it's not exhaustive, but just to try and codify it in an easy way to view, we represent it this way. So our total committed investment to date is over 45 million, and some of that is already completed and some of that is ongoing. One of the things we like to highlight, because it speaks to some of the things I think Barry will get to later, is our Vaccine Analytics and Assays Center, which is a physical space. So it includes a suite of instrumentation. Whoops, let's go back one. There we go. It includes a suite of instrumentation for characterization of a range of key attributes of vaccines. So it's not about manufacturing them, it's more about characterization. And these could be things that range from aspects that tie into what you would maybe glean from the prior slide, which are around potency, stability, adventitious agents and sterility, physical or chemical properties, things that have to do with solid state properties and beyond. And it would be across different vaccine modalities and sample sources. The focus here is as a test bed. We're not intending that this would be as a contract testing laboratory. So it would be a sandbox of sorts. And we've already invested quite a bit in this space. We've been doing a lot of work there, but it's a physical space with some of the instrumentation is ready and available once we kick it off. And so we're going to be doing that with pilot projects later this year. And we'll put a little plug in for those who might be interested, depending on who is in the audience, for joining NIIMBL and working with us on this from an employee perspective. We're always looking for highly qualified scientists and there's a typo there, I apologize, to join our team, either as an employee or as through member participation. So I think with that, now we're ready for this. I'm going to hand this over to Jen Mantle. Jen, I will run the slides and you just let me know when you want to advance things.
Sure. Thanks so much, Chris. I'm excited to share an overview of our efforts in next generation sequencing and how they can help contribute to our vaccine program. Next slide, please. So next generation sequencing, or NGS, is a technique that can be used to identify DNA and RNA in a sample, which has a lot of potential applications in biopharma manufacturing for all product classes, not just vaccines, but they can be used for vaccines, therapeutic proteins, and cell and gene therapies. And so NGS can be used to characterize host cells. It can be used to characterize products such as viral vaccines or mRNA vaccines. So for example, a recent publication described using NGS for the assessment of genetic heterogeneity as a lot release test for a new oral poliovirus vaccine. NGS can also be used for detection of viruses in a biopharmaceutical manufacturing process. So in the figure Up top, you can see viruses can be introduced at virtually any step of the process. They can be present in raw materials, or they can be introduced by the operators or the environment along the way. And so to ensure that the final product that we put in patients is free from unwanted infectious virus, we have to do appropriate testing and risk mitigation. So first, we make sure that our starting materials are selected with a low risk of contamination. We do testing of cell banks, in-process materials, and final product. And we also incorporate steps into the process to inactivate and remove adventitious agents during purification. So a recent publication out of an MIT consortia provided insight into common viral contaminants in biopharma manufacturing and the impact of these events. And others have shared their approaches for using NGS to make risk-based decisions when bringing new materials into a manufacturing plant to minimize the risk of costly shutdowns and cleanings. So this approach was specifically used for making COVID antibody therapies, whereas speed was one of the most important drivers for the team. So while the industry generally has had an excellent safety record, viral infection is still a real risk with severe consequences such as drug shortages, millions of dollars for investigations, cleanup and corrective actions, and also altered public perception. So while there's many different NGS technologies that can be used for adventitious agent detection, the general workflow involves taking a process sample or a raw material sample that may or may not contain viruses, extracting DNA and RNA from the sample and creating a library. And then it's run on a sequencing instrument, and then a bioinformatics pipeline is used to identify the viral DNA from the background. Next slide, please. So NGS is a promising technology for reducing animal testing and vaccine manufacturing. So this table compares NGS to classical in vivo testing as well as PCR-based testing. So in animal tests, generally speaking, after the animal is exposed to a sample, it's monitored for clinical signs of viral infection. PCR tests, they amplify and detect specific viral genetic sequences, so you get what you're looking for. And NGS has the potential to identify all genetic material that's found in a sample. So with respect to detection of viruses, detection of different species of animals are only able to be infected by certain viruses, so there's a detection limitation there. PCR is pretty good, and it can be multiplex, but it's a targeted approach and you only get what you're looking for, while NGS can theoretically detect any known virus. So we say that it has broad detection capabilities. PCR and NGS are both pretty specific and can identify a virus present in a sample. However, if an animal is present for viruses, we would have to do further testing like PCR or NGS to identify which virus was present in the sample. Sensitivity of animal testing is generally pretty low, and turnaround time can be about 6 weeks or more, with about 4 weeks dedicated to observation of the animals for clinical signs. But the sensitivity of PCR and NGS are generally better than animal tests, and turnaround time is on the order of 2 weeks, which is a big improvement over 6 weeks. And so in this last row, one advantage that animal testing over molecular-based methods has is that in vivo tests do detect replicating viruses, while molecular methods detect the genetic material of viruses only. So for simplicity, we don't have cell-based tests listed in the table, and they are an important test in the suite of tests for adventitious agents, but they share similar limitations in terms of detection and specificity and turnaround time with the animal tests, but they do also reduce the number of animals used. So molecular methods like NGS present a solution to some of the main challenges of traditional virus detection methods, but the technology isn't quite as mature, and there's still questions about how NGS compares to traditional methods. Next slide, please. So to address some of these questions about how they compare, there's some ongoing work within NIIMBL around using NGS for adventitious virus detection. So NIIMBL is a participant in the PDA Adventitious Virus Detection Interest Group, which is working on interlab spiking studies for different NGS technologies, among other things. NIIMBL has also supported a project shown here in this space through the Global Health Fund project call, and that project's called Evaluation of Next Generation Sequencing as a Rapid Alternative Assay for In Vivo and In Vitro Adventitious Virus Testing for Vaccine Safety. So this team is led by Dr. Arifa Khan at the FDA, and their project partners are GSK, Millipore Sigma, and the University of Delaware. So the first aim of the project is to compare NGS-based methods with in vitro cell culture and in vivo animal assays, and also PCR. And so the team is working on the wet lab protocols, implementing those protocols in different labs, and looking at the limitations of the different methods using the exact same samples. And the team's also working on the next iteration of the reference virus database, or RVDB, so that we have an accurate interpretation of NGS data. And they're also working on the analysis pipelines. And so this project, when completed, will have a number of impacts, most importantly, the potential for reduction in the number of animals that are needed for adventitious agent testing. The project will also result in the streamlining and standardization of bioinformatics pipeline. And hopefully it will reduce the cost and time of adventitious virus testing, leading to more rapid product development. And so far, the team is making really great progress, and the project's nearing completion of this portion of the project, and the NGS assays and analysis are complete. The in vitro and in vivo testing is nearly complete, and the RVD updates are underway, and they'll be completed by the end of the project. Next slide, please. And so to further advance NGS assays to reduce animal testing for vaccine manufacturing and for other applications in biopharma, NIIMBL has built what we're calling the NGS Test Bed at NIIMBL headquarters in Newark, Delaware. So our vision is that the test bed will support accelerated decision-making, reduce animal testing, and characterization of emerging biopharmaceutical products. So to that end, we've acquired a number of different sequencing platforms so that assay can be developed on the most appropriate platform, or so that comparisons between different technologies can be carried out. And so we want this resource to help increase comfort with NGS within our community by generating data and making that data widely available so that it can be referenced by others. And so we've acquired a number of different sequencing platforms, starting with the Thermo Fisher 3500, which does Sanger sequencing, which is capillary based and low throughput, but highly accurate. Then we have the short read platforms. There's 3 of them. Each of them have different sequencing depth capabilities. They're all from Illumina. There's the MiSeq, the NextSeq, and the NovaSeq. And they all generate reads that are about 150 to 300 base pairs long. Well, these sequencers vary in runtime, cost per run, and amount of data generated. So we get about 25 million reads for the MiSeq, up to 20 billion reads for the NovaSeq, which is roughly the equivalent of 40 human genomes that can all be run at the same time, which is pretty impressive. So these short read sequencers are the most commonly used, and they're good for accumulating a lot of data. And then lastly, we have 2 long read platforms. So there's the Oxford Nanoport, which generates reads that can be 10 kilobases to ultra long 300 kilobases. It's ultra portable. The smallest ones are about the size of a Snickers bar, and it has pretty low accuracy. There's some challenges with interpreting the data, but it has certain use cases that make it the most desirable choice. And then we also have the PacBio Sequel 2E, which generates reads that are about 10 to 25 kilobases, and they're pretty high quality. And so we've also incorporated some automation into the test bed. So we have automated nucleic acid extraction, and we have automated library prep instruments. And we've also supported some of the bioinformatics infrastructure required to store and analyze the data generated by the sequencers. So within NIIMBL, this NGS test bed is supporting activities for the vaccine program, such as adventitious virus detection, as well as host cell characterization for our process intensification and big data programs. And we're supporting product characterization for emerging products programs as well. And I'd also like to share that it's always been our vision that the NGS test bed will be available as a community resource, and we're working through the details on that, but we anticipate making that available to our members later in 2023. So if you're interested in working with us, please don't hesitate to reach out, and I'd be happy to connect with you. All right, thanks, Chris. Back to you, Barry.
Yeah, thanks, Jen, for your very nice summary. And so we're very interested in this whole issue of better analytics for vaccines. Clearly, as Jen described, it would reduce release times and improve safety for not only vaccines but other types of products. And the improved analytics for characterization of a vaccine will help manufacturers by enabling bridging studies as they go up in manufacturing scale. As a company needs to move manufacturing from one location to another, that also requires bridging studies, and if that can be done analytically, that's a great advantage to the company. And also for process improvements, when making a vaccine and the development teams come up with ways to improve the process, then better analytics allows, again, bridging from one process to another in terms of comparability to make sure that the vaccine will still be efficacious and safe. So we're very excited about getting these resources for the analytics and assay lab and also the NGS lab. So toward the end of the year, these should be running. We're starting to identify members who wish to participate, and this webinar is part of that, in a way, the first part of that process, getting the word out and finding out who's interested to participate. We'll certainly be reaching out to our major company members to get their engagement. We want to form a steering committee with SMEs from large companies. Some of you will have listened to the viral vector program, and of course there's some overlap, so we want to integrate our plans with the NIIMBL Viral Vector Delivery Initiative. Chris mentioned we just approved 3 different projects related to improved potency assays for messenger RNA, and that's the project called 6.1G. So we're excited about that. So we definitely want to be tracking progress as the groups proceed on improved potency assay for messenger RNA. And then finally, Chris and I are involved in a collaboration with other groups like PATH and NIIMBS, Bill and Melinda Gates Foundation, CEPI, which is really to the question of critical reagents. So when there's a pandemic, one of the rate-limiting steps early on is to develop key assays, key potency assays, for example, and these require custom-made reagents. So the development of those critical reagents with a new pandemic is really central to the timeline. And so we're eager to continue contributing in the way we have been, including we funded projects in this area, but also to continue this as we go forward. Go to the next slide. So the next steps, how can our members contribute? And as has been emphasized, NIIMBL is a member-driven organization. We want to hear from you. We chose the general topic of analytics and assays based on feedback from all our company members as a good area that every company is interested in, and in some cases willing to collaborate to improve methods in this area. And so choosing this came after a lot of discussion. And you can help us, for example, a company that's on a project providing relevant samples is really extremely valuable to such a consortium. Helping us staff our lab work at the NIIMBL headquarters would be both a career opportunity, workforce development opportunity, as well as great for NIIMBL. So that would be another way. Certainly, members of the steering committee will be seeking guidance on the future direction of vaccines at NIIMBL and monitoring progress. We'll have discussions both between academic members and industry members, the relationship, and government members, the relationship between structure and function. And finally, reviewing the data we're generating. We have a lot of initiatives ongoing as we speak, and creating publications. But of course, for any company that's actively involved, you get an early look at all this work, and also, publication tells you so much, but some of the know-how behind the methods will be developed as part of this activity, and that is not always clear within a publication. So we're excited about future. We built this fantastic resource, and thinking about next steps. So I will pause at this point, and we'll invite questions. And Chris, maybe you could repeat the instructions for
Yes, good, great
questions.
point, Barry. So yes, so a few people have already taken the chance to pop a question that came into their mind into the chat function in WebEx. Depending on which type of computer system you're using, that chat function may show up in different places. I do not know why that went there. We want to go back here. So we're taking questions now, and then we'll have a couple of sort of just simple updates and announcements at the end. So if you can pop your questions in the chat, we'll give people a minute or two to do that. We'll take some of the earlier ones, and then if things are in a similar vein, I'll curate them and combine them so the same question isn't answered, doesn't need to be answered multiple times. And I can pose them to either Barry or Jen, or if it's one that's appropriate, I'll take it myself. And some of them would be ones that we may not have time to get to today, and that's okay. But you'll see at the end there are ways to connect with us, and we can follow up offline, maybe via email. In some cases, questions that are really more to an individual are probably better left as an individual contact with us rather than trying to do this within a webinar format. All right, so let's see what we have in here that we can start with. So there's a question here around, and this would go maybe to you, Jen, for NGS adventitious virus detection. Is the strategy limited to or focused on identifying target virus, or does it also focus on giving a positive signal when a non-host nucleic acid is present?
Yeah, that's a good question. It can be either. So we can do a targeted approach where you look specifically for sequences that you're interested in, perhaps pulling from that reference viral database. Or you can do more of a shotgun approach where you detect and flag any nucleic acid sequences that are not part of the host. And you might imagine that there's different bioinformatics pipelines that need to be established for both of those approaches.
Great, thank you, Jen. So one that's going towards the vaccine analytics test bed side, which probably I'll take this question. Well, there are a couple here that are essentially in the same vein. So it's asking around, or the question is basically focused on, would this be around sending samples? Would it be around sending people? How's the work going to be, shall we say, staffed? And so those are all great questions. The short answer is yes to sort of all of it, in that there could be a question of submitting samples, but they need to be industry relevant samples towards something that would probably be more concerted effort. Again, this will be a test bed, not a contract testing laboratory. And then we will have our own staff that will be there to help basically run the infrastructure, but we're very open and encouraging to having participants who are, for example, subject matter experts from our members, and that you would need to be a member to participate in this. But they could be seconded for a short period of time, whatever short might be, and whatever makes sense within their other duties, so that they don't need to come in and operate the equipment as much as they would be there to make sure that the relevant expertise and background knowledge is there to make it most useful. Hopefully that covers that. Let's see. There's some here that are probably better done offline. If one's thinking about contributing as a steering committee member, I think that would be best to email to us separately. And that's a good one-on-one discussion, because it could look very differently for different people. So here's a different one. Let's see. Oh, is the focus, and I think they're alluding to the analytics test bed, will be mostly focused on mRNA or viral vector vaccines. It's not or, I think you would say it's at least and, but a lot of the technology that will be in the analytics test bed will be applicable to other modalities. So I think we're still open there and still looking for where the steering team chooses to take that. Let's see, what else do we have in here? Question around will the actual list of instrumentation that's in the final analytical lab, the sandbox, will it be available publicly? I think eventually it will be. At the moment, it's still being built out, so it's a little early for that. Certainly will be available for all members to have access to. Let's see. Yeah, there's some here that are more specific to the individual who are asking about availability of space and whether we would want to collaborate on something in the future, or people would have access to things that either formerly were at the UD site and in a different facility that's not actually NIIMBL, or that has to do with space that might be available locally. Please reach out to us separately with those questions. Those are probably not so simple to answer in a webinar format. So then, Jen, here's a related question over to you. I think it's asking, do we know whether the relevant agencies will be open to leveraging NGS to replace the existing adventitious virus panel for release testing, as an example?
Right, that's a good question. So I don't work at the FDA. I cannot speak for the FDA or any other regulatory agency for that matter, but I do think that there is a lot of enthusiasm around looking at these types of technologies. And I think that everyone can get on board with reducing animal testing. So I think that there's an openness and a willingness to take a look at data.
Okay, thank you, Jen. And I'm going to kind of combine a few here into a similar theme. So essentially, is there an interest? Is there basically an appetite for having what we're looking at here or other resources? I think they mean for around, I think the way the theme is going is around characterization of late stage vaccines that might have been developed by someone else but are now available, and could then, what we're working on at NIIMBL, would those resources be available for characterization, as an example, only for members or perhaps for all? Or would we be looking at something else that might be, for example, sharing what could be done as characterization, but at least who's vetted by NIIMBL? I think it's a great question. I think the initial stages of what we're trying to do would be that eventually what we work out with the NIIMBL members in terms of characterization certainly will not be vaccine specific. This is not going to help get one medicine over the line. That's not what we're in a position to do. But what we may develop in terms of, for example, protocols, best practices, what have you, these don't need to be peer-reviewed publications. They may be white papers. They may be something even more practical than that, would ultimately be shared publicly, but would be developed with the input and the subject matter expertise of our members. And of course, that would give them, shall we say, the first glimpse into all of that, and also a lot of influence. But if this also benefited the larger community by helping to get some of these, maybe as you said, as I summarized it, late stage candidates that maybe weren't going to go any farther, it helps another portion of the community get that over the line to get it to patients. We certainly would love to see that as well. Let's see, we've got some more coming in. The problem with the chat is that you got to make sure to scroll back and forth. There's one here that is effectively asking, would with the applications, or a couple actually coming in saying that with the applications of what NIIMBL is focused on, also include things towards food products. At the present time, I would say that's a good question to have as an individual organization with NIIMBL, especially if you're coming from a company, for example, that would potentially join, because we should talk about where the boundaries are in that space, the way that the Manufacturing USA institutes are set up, and the way each of them have their own mandate. There is certain boundaries we're not allowed to stray from with respect to the kinds of applications. To date, we don't have any companies, I would say, that their primary focus is on food products, but we can have a separate discussion with that offline. So I would say you reach out to us and we would get you in touch with the right people. Let's see, am I missing any? It looks like we're slowing down on the new ones coming in. I apologize for anyone if we didn't capture it exactly the way you had in mind. Again, you can follow up with us separately, and we'd love to have the engagement. I think that's where we are, Barry. So if you have other things you'd like to add to any of the comments we've made, please do. Otherwise, we can do a wrap up.
No, I think covered it very well, Chris and Jen. So yeah, we can go to the wrap up slides.
All right. So for those folks on the line who are members or looking to become members, once you would become a member, you can see more than just a lot more than what we've shown you today. I mean, we listed out in one place, this is the number of projects. We didn't give you any detail except for the lovely example that Jen Mantle took us through. But there's a lot more there. And if you want to learn more and also get more engaged, once an organization becomes a member, any employee of that organization is also a member, and you'd have the ability to go in and self-select for yourself within our NIIMBL portal, within the confidential portion. And you can see quite a bit of detail about all the different projects, not just the ones that may be around the vaccine manufacturing program or the larger vaccine umbrella. It also spans a number of different areas. So if your organization, if you already are a member, but you didn't realize it, you could reach out to NIIMBL and we can make sure to get you an account. And if you are not yet a member, once you would become a member, then you would have that option. And you can actually follow, quote unquote, follow it. Literally, you'll get routine updates for just the projects you're interested in. But you do have to be a member or a federal stakeholder. Otherwise, all you can see is a much smaller amount of information, which is on the public-facing website. Also, for those who are members or looking to become members, we have our upcoming national meeting in late June in Washington, D.C., a beautiful venue. Those who are members may have already seen information on this. Those who aren't, this would be an opportunity to also engage in a face-to-face. It's really a great program that's shaping up. However, it is only open to members and federal stakeholders. So I think with that,
Yeah, and it does indeed include some vaccine topics.
It does indeed. So if you just visit the NIIMBL website, the public-facing one, which is just www.niimbl.org, then you would be able to find contact information for, particularly for non-members, for being able to get in touch with the right people. Just basically contact us, and you would be able to. We'll get you in touch with the right person, depending on where you're coming from, academia, industry, different federal labs, etc., for how to get access to things. So I believe that is my last slide. I did see something else just pop into the chat. Hang on a moment.
And while you're doing that, Chris, worth mentioning, this webinar was recorded and presumably is available for future access.
That's right. It'll be, for those who fill out the survey, you'll get almost immediate access. And so that last one, is there an agenda for the national meeting? It's not been published yet. It's a great question. We're working to lock that down, and it will be posted soon. Oh, thank you for one of our support folks who already responded to that in the chat. So I think we'll thank everyone for their time and attention today, and we'll close at this point.
Yeah, thanks for your attention, everyone. Look forward to working with you in the future. And thanks, Jen and Chris, for a very clear overview. Thank you.